Good morning and welcome to this talk on denoising techniques. This is part of the ISMRM educational session, fMRI across spatial and temporal scales. I have no conflicts of interest. To understand noise, let's reflect what functional MRI is about. We're basically acquiring a movie of the behaving brain, and then the analysis stage answer the question, are there any changes in that movie that are related to our research question? So changes that might be correlated with the task, or for resting state fMRI, changes in different brain regions that are somehow synchronous or correlated. Now, while this question is simple, the answer just looking at this movie might be difficult what this subject is doing. You might argue, well, I'm not interested in the time series per se, but only in the fluctuations, so I could subtract the mean image from this time series. If you do that, you see a lot of fluctuations popping up. However, whether there are any meaningful ones or functional ones might still be hard to detect. So you see some fluctuations at the borders of the brain that might be due to motion. You see salt and pepper fluctuations inside and outside the brain, that is thermal noise. And you see also fluctuations within the brain that might be physiological, but not necessarily neural, like here in the ventricles. However, spotting the actual functional bold effect here might be hard. fMRI usually undergoes a lot of image processing to improve the situation, so some alignment against motion and some spatial smoothing to remove thermal noise. And in the end, you might get a time series like this and might be able to spot a bold effect here in the auditory cortex. But you also see a lot of fluctuations in the ventricles. So noise is a constant companion in our fMRI analysis. What we learn from this exercise is that in order to identify fMRI signal and noise, we're typically looking at regional fluctuations in the time series. So one could argue that the localized time series is the fundamental information unit of fMRI. And if we look at, for example, one voxel in the brain in this time series, we typically distinguish signal, which is fluctuation through functional contrast of neuronal origin, like bold, from noise, which is all other fluctuation in this voxel. However, in practice, these two are superimposed. So after we measure, we might not be sure whether there's really an effect in the data because it's noisy. And this is indicated by these shaded areas in this time series. In actual fMRI experiments, the situation is even worse. So there's a lot more noise than signal change. And in order to do any meaningful inference on effects in the brain, we typically have to assess the ratio of the two. And any meaningful model has therefore to estimate both the effect size and the noise power. And their comparison usually assesses something like statistical significance. If you look at an f-test or a t-test map in fMRI analysis, they're typically ratios of an effect and a noise estimate. The goal of denoising can therefore be defined as trying to improve the sensitivity of your fMRI analysis by reducing uncorrelated noise floor and boost SNR. But there's also fluctuations that are correlated with your effect of interest. And a second goal of denoising is therefore to increase specificity and reducing any signal correlated noise. In the remainder of this talk, I'm trying to give an overview on identifying noise and explaining noise origins for fMRI, and then go through different stages of denoising that will help you reducing noise in your time series. And I will conclude with some best practices. How do we identify noise? You've seen one example now, just by looking at the raw data, movies of the behaving brain. This is probably the best way to look at noise and identify it, but it's cumbersome. So in practice, we rely on quality control measures to automatize this process and to quantify denoising performance. For example, we can look at spatial temporal characteristics of the voxel time series. Is there any frequency that is suspicious or any regions that are typical for noise, like the ventricles we've seen? There's a very elegant way to look at raw time series, which is called the gray plot, that basically orders all voxels of the brain in the gray matter and then in the white matter along the y-axis, and then puts the time series as an intensity plot on the x-axis. And here you can see the example of head motion of about 1 millimeter and how it changes the intensity of your voxel time series in most of the gray matter. You can quantify noise by statistical images, and the most common one is signal to fluctuation noise ratio, SFNR, or also temporal SNR, TSNR. And basically, you take the mean of a time series of voxel and divide it by the standard deviation of the time series. So you get one value per voxel, and you can create a map out of that. If you have a model of the noise, you can have a look how much variance in your data is explained. For example, with an f-test, you see the example of cardiac physiological noise. Or you can look at how assumptions about your noise are violated, for example, the Gaussianity of thermal noise. And the non-Gaussianity measures like kurtosis or skewness give you an indication of artifacts. Finally, if you have a noise model, always look at the residual mean squared image of that model, which tells you where your model didn't behave so well. So it could be the model of the task or of the noise. If you look at the residual, you see that there are certain areas in the brain where you have missed some important fluctuations that you cannot explain. Where does this noise now come from? You know that in fMRI or an MRI in general, we don't acquire data in image space, voxel by voxel, but we acquire it in the Fourier space, in the K-space of spatial frequencies that are linked via the Fourier transform. Very generally, we can say that anywhere in this process, be it the object itself, or the encoding via magnetic fields, or the detection via receiver coils, we can have noise entering our time series. And this noise stems from two main sources. One is the MR system, the scanner, and the second one is the image subject. And unfortunately, each of those can affect all these entry points of noise. One example for thermal noise is that there's ions in the subject that move around, but there's also electrons in the coil that move around, and they both create thermal noise. In practice, the generating effects and the mediating mechanisms that lead from these noise sources to the actual entry points of noise are manifold. And we'll go through some of them over the next slides. Just to give you an example how to read this slide, you can see how the noise source, the MR scanner, generates noise via heating. By heating, for example, you have a high duty cycle in fMRI, your system heats up, the magnet is deformed, it expands, and the gradients get softer. And all of this leads to changes in the magnetic fields and ultimately in your image time series. Now let's turn to the subject as a major noise source. Head motion is one of the noise generators. So when the head moves, you have a rigid body displacement that changes the magnetization. This can be sudden motion, head jerks, and slow drifts sliding out of the coil. So the range is usually millimeters or degrees of rotation, and the spectrum accordingly between 10 Hertz and 1/100th of Hertz. What is important to note is that even if you align your images after the experiment, this preprocessing cannot correct spin history. So a very simple example is if you have movements between the slices, you have this rigid shape here, subjects move back and forth, very unlike the rigid body brain you had if you didn't have any movements. And there's also small partial volume effects related to that. And think about the spin history. If you think you have several excitations, several TRs, and you assume a head that is basically steady, then the same slice is excited at the same time with the same time intervals, and you have a steady state magnetization. However, if you have a movement between the slices, it means that some parts of that slice are only excited once instead of twice, or once instead of 3 times, or and that means that you have a fluctuation that is related to the movement, but doesn't stem from any neuronal origin. And there's also another pathway that enters via the encoding magnetic fields, because if you change your pose in the scanner, the susceptibility also changes. And you can see this in the phase images here. This leads to changes in the B0 maps, basically, and local distortion, especially in the orbitofrontal and temporal areas. So you have deformations that are nonlinear and local that change with the movement. The cardiac cycle is also a potent noise generator that mainly enters through the magnetization point. There's effects that are directly related to the cardiac phase, to the heartbeat. They're on the order of 1 Hertz. And they are located close to vessels and CSF flow, as you can see here, the basilar artery and some F-test of cardiac noise. The mechanism behind that is that even though the skull is a rigid body, during every heartbeat systole, blood is pumped into the brain. So the vessel volume increases. And because the volume overall cannot increase, the CSF is pushed down through the cerebral aqueduct in the 4th ventricle into this spinal cord. And you can see this rather large effect, this displacement here in the aqueduct itself, but you also see effects because in the major vessels, for example, here in the ACC, in the basal artery. And during diastole, this flow is reversed and you have CSF returning into the brain. There's also an effect of the cardiac cycle on a much slower scale, which is related to heart rate variability. So it's only 1/10 of Hertz and is widespread in the gray matter. The mechanism is not completely clear, but it's assumed that the blood pressure and the vessel tone changes cyclically. And if the blood pressure increases, the blood flow increases, and cerebral blood flow, CBF, is one component of the bold signal. Also, the breathing cycle can act as a noise generator. Again, synchronous with the respiratory cycle, which is typically 15 to 25 breaths per minute, so that relates to 0.4 hertz. And typically, this is a global effect and occurs also at brain edges. And the mechanism is that within the breathing cycle, the chest and also the head moves. And this, of course, creates direct motion of the head. But there's also a pathway through the encoding magnetic fields, because the changes in the lung volume change the encoding magnetic field. And for EPIs, usually this results in a shift in phase encoding. And that's why you have these major effects here at the edges of the brain. There's also an interaction with the heartbeat, because the heart rate increases during inhalation. That's called respiratory sinus arrhythmia, and is mostly visible in the aqueduct or in lower brainstem regions. The second, again, slower effect of the respiratory cycle relates to the respiratory volume per time. It's on the order of 0.03 hertz and widespread throughout the gray matter. The mechanism here is again related to the blood flow, because breathing rate and volume alter the arterial CO2 levels. And since CO2 is a potent vasodilator, you can see that the increased blood flow again leads to increased bold. How can we actually reduce the noise that is generated by these different mechanisms? I would like to refer to the waste or noise hierarchy here, which has been invented for recycling, but applies to denoising and fMRI as well. And the key message here is not injecting noise is better than removing it afterwards. So whenever you can avoid noise, do so. And reducing it and reusing it are the second best options. Avoiding means you change something about your acquisition, reducing during the preprocessing. Reusing is co-modeling of noise and signal. To avoid noise during the acquisition, let's focus on the two main noise sources, the system and the subject. It's easy to adapt sequence parameters for reducing noise. For example, if you have a high spatial resolution, you reduce partial voluming, so you mask out noise locations like the ventricles from your gray matter areas of interest. Similarly, if you have a high temporal resolution, you might be able to filter out parts of the noise spectrum, for example, the heartbeat, that you're not interested in. And there's 2 lectures in this session that talk about these two applications of high spatial resolution and high temporal resolution. Also, if you need neither of them, consider decreasing your parallel imaging or multiband acceleration, because this will reduce thermal noise. A second level of avoiding noise during acquisition is adding additional sequence elements. You can add some extra acquisition lines, navigators, that can help you to identify breathing artifacts related to the magnetic field, or also head motion to a certain extent. And for a more localized approach, you can actually export phase images, because the MR signal is complex, and not only the managed images can be used for MRI analysis, but also phase data can give information. There's also acquisitions that acquire data at multiple echo times that can be used to disentangle signal and noise, because the bold signal specifically is dependent on the echo time, whereas others effects like motion are not. And the harder part for the acquisition stage is to include prospective updates, real-time updates to your noise. For example, you can trigger your acquisition to the cardiac cycle to remove cardiac noise, or in space, you can update your slice geometry or your fields to observe motion or field fluctuations from the breathing cycle. What shouldn't be underestimated, though, is that the subject also plays a major role in reducing or increasing your noise. So careful instructions, so that they should remain still, or that they shouldn't breathe with a paradigm, are important. And you can even provide tactile feedback to the subject with a tape over the forehead. And a very comfortable option is to have stabilizing padding or head cases, which now can be printed individually in 3D for each subject. So that's probably the best option to keep them still if you don't have prospective correction. And again, be reminded that instructions also help with the breathing cycle and not correlating your task to the respiration. After we have finished the acquisition, signal and noise are inevitably mixed. So denoising or reducing noise during preprocessing is always the task of disentangling the two. which can be sorted in 3 different stages. So we first have to find a separation criteria to identify noise, some prior information like temporal or spatial characteristics that we mentioned, or some independent measures. We can have peripheral recordings of physiology or of motion. The second stage is to model the noise components, and the third one to reduce your data, basically removing the modeled noise from it. Here's an example for this pipeline for motion reduction. So if we have had motion in our time series, it's translation and rotation because the head is a rigid body. And the separation criteria could be related to our image processing. So we have realigned our data, and then we get parameters from that, rigid bodies realignment parameters. And priors can be related to some assumptions of the effects of this motion. So in the temporal domain, we assume that we have correlations to the estimated motion that are immediate because of the displacement. So there's an immediate intensity change if we have a displacement, but there's also the mentioned spin history effects. So they might be delayed, so motion or might have an effect in a later volume of the time series, or there might be nonlinear effects because of the spin history. And spatial priors are, for example, that motion effects are usually close to tissue and edges because there's a high intensity drop. Noise modeling in this case can be linear. So we can just use the realignment parameters in a general linear model, or we can model the delays via the derivatives of these realignment parameters and some nonlinearity via the squaring these parameters. Second way to model, still a linear way, is censoring. So we can inform our GLM of sudden motion spikes, for example, by thresholding the frame-wise displacement, which is depicted here. So we take the sum of the absolute value differences of these motion parameters, and then we see certain spikes here, and we can basically censor or scrub this volume by creating a stick regressor that is 0 everywhere, but 1 for this volume. So this volume will be completely ignored in the general linear model. Its variance will be removed. The actual noise reduction using a GLM then works via a confound matrix, X0, from which you can basically derive a residual forming matrix, K, that makes sure that you remove everything that can be explained with this linear model. That's basically a noise model, but retains everything else. And what is important here is that the statistical analysis you afterwards perform on that filtered data has to also respect this filtering. So also your effects of interest should be filtered with the same matrix. Otherwise, you introduce anti-correlations between the physiology and your task. Let's continue with this conceptualization for physiological noise. Here we can distinguish 2 classes of separating signal and noise. One is the exploratory denoising class, and the other one is the model-based using peripheral recordings. For the exploratory data-driven denoising techniques, we basically use the image data on its own, maybe together with an anatomical brain image, and we rely on priors. It could be temporal priors, for example, that the breathing and cardiac frequency on a certain range. It could be spatial priors, for example, that physiological noise is close to CSF or vessels, or white matter doesn't have a bold signal. And it could be the combination of the 2 in training data. So we can have a certain population where we manually identify physiological noise and create a dictionary from that we then use in a different cohort. The noise modeling itself relies on decompositions of data that make different statistical assumptions. So one popular one is principal component analysis that takes data from a region of interest that is assumed to only contain noise, like CSF or white matter, and then identifies orthogonal sources in that region, basically time series of maximum variance that are then used for regression. And the second one is independent component analysis. Here we assume that the noise sources are statistically independent, either spatially or temporally, which doesn't necessarily mean that they're independent in their physiological causes. So there can be several components that all relate to the cardiac cycle, for example. And there's global signal regression, which makes the least assumptions and just says that the mean over all brain voxels also gives you a time series, and that's basically noise and can be regressed out. And then actual noise reduction, again, is simple. We take these identified noise components and subtract them from the data. So here you see the example of the ICA, and as you can see here, there's a certain component that is strong in the sagittal sinus, so major vessel, and it has prominent frequency at about 0.03, 0.04 hertz. So this might be related to the heart rate variability. For the recording-based denoising techniques, the most prominent example is the retrospective image correction, retro-ICOR, where you acquire some data from the heartbeat, which is usually a pulse plethysmographic unit, pulse oximeter, and some data of the breathing cycle, usually respiratory bellows. And then you pre-process these physiological readouts to arrive at a representation of the cardiac phase and the respiratory phase of your physiological cycles. For the cardiac phase, you look at the distance from your acquisition time point from the last heartbeat and divide it by the distance between two subsequent heartbeats. And for the respiratory phase, you look at the amplitude of the respiratory bellows signal during your acquisition compared to the maximum inhalation and maximum exhalation amplitude of that signal. And then actual noise modeling then is a Fourier expansion, which assumes that you have a linear effect of these cycles. But because you model both cosine and sine terms, you allow for a possible phase delay. So different slices could have a delayed response to the cardiac or respiratory cycle. And the second important thing is that you also include higher order terms in this Fourier expansion, so n times the phase, to avoid aliasing. Because when you actually sample a cardiac fluctuation that occurs at 1.1 hertz and with a TR of only 2 seconds, it actually looks like a very slow fluctuation here in blue because your frequency has been aliased or folded into a different part of your spectrum. And that means that you also have to model these higher order effects, otherwise you miss these apparent slow effects of the cardiac cycle. And in the actual noise reduction stage, you just take nuisance regressor, so you sample these Fourier expanded phases at one time point per volume and then put it into generally a model, as you can see here. You do this for a group study and look at where in the brain the cardiac and the respiratory and the interaction components explain variance. You see basically that the respiratory components expend a lot of noise at the edges of the brain, whereas the cardiac is more localized here in the ACC, a lot of brainstem areas. The interaction is also in the brainstem, in the aqueduct, the cerebral aqueduct, and here also around the brainstem. You can also see, for example, here in the insular cardiac effects of retro-ICOR. Heart rate variability also uses this pulse oximeter signal and pre-processes it to compute the heart rate in a sliding window, and then models the effect of the heart rate on the brain as a linear time invariant system. So in the original work by Chang et al., they tried to estimate a cardiac response function by explaining the maximum variance in gray matter that can be reduced by using the heart rate and this linear time invariant model. And if you convolve then this heart rate with this cardiac response function, you end up again with a regressor that you can regress out. A similar approach has been done for respiratory volume per time. So again, you take a respiratory bellows and you pre-process it to compute an envelope on the actual respiratory time series. And this is this respiratory volume per time, which has this peak frequency of 0.03 hertz, which is somehow related to end-tidal CO2, so to the vasodilation. Again, if you put in a linear time invariant system assumption, you can estimate a respiratory response function, again, with a peak and then an undershoot and a slow return to baseline in about a minute, and create a regressor from that enters the GLM. The trouble with using all these noise models in a pre-processing step is that effects of interest can have both neuronally induced, so functional, and also physiological consequences. So think about arousal that changes your heart rate, interoception, which might change your breathing, or think about the neural centers of the autonomous control in the brain that of course also show functional activation, but they're also related to how that changes the physiology downstream. So noise reduction as a pre-processing step might filter out too much. So for example, here you see a resting state network from the literature, and the clean data shows it very clearly, but also the removed, so basically the putative noise part that was removed, if you look at this, also shows such a pattern. And if you don't do your modeling carefully, as mentioned before, you can also use anti-correlations if you use denoising in the pre-processing. The solution to that problem is co-modeling for denoising. So this means you handle the signal and the noise simultaneously, and the simplest approach is to include your nuisance regressor in the task GLM. So instead of having two different models, you have one. Here you have the example of a block design with on-off stimulations, some cardiac, respiratory, and interaction retro-icor regressors, and some realignment parameters. There's also more complicated models like state space models, or using cross-correlation test-retest reliability for your task to also co-model signal and noise. For most of the methods that were introduced as pre-processing before, this co-modeling is applicable and recommended when possible. However, the integration is difficult if you model your effects of interest very differently from your noise. So if you have a multivariate ICA, but then a master univariate GLM, it's hard to integrate the two. And of course, if you have also nonlinear noise reduction. Let's conclude with some best practices for denoising. First of all, this was a very broad overview, but I just want to say you don't have to reinvent the wheel. Toolboxes exist for all these discussed denoising techniques, both for the external recordings, Retro-icor, HRV, RVT. All major software packages for fMRI analysis, AFNI, FSL, SPM, have some toolboxes for noise correction. And also the data-driven approaches, ICA and also anatomic component correction, are available in a lot of packages. In terms of best practices, the first thing is to think hard about your experimental design. So does my task correlate with physiology? And can I instruct my subjects, or can I buy some special foams that keep them still? Equally important, think hard about your acquisition parameters. What spatial temporal resolution can I afford? How does it help my denoising? How is it important for my research question? Acquire physiological recordings, at least for pulse and breathing whenever possible, but also check and improve the recording quality. It only takes a minute to look at whether the respiratory bellows is attached or not. And whatever you do, do some denoising, at least of motion physiology. It can be data-driven, can be recording derived. Most importantly, do some quality control. Look at your data a lot, before and after the denoising. Is it effective? Is my denoising too aggressive? And scrutinize both the signal and noise, so don't just throw things away without looking at them. This completes the noise hierarchy by a fourth item besides avoid, reduce, and reuse. Because in the actual waste hierarchy, we have the recycling as the fourth component. And what this means for fMRI is that one scientist's noise might be another one's signal. So these physiological networks that we identify with denoising could in itself be meaningful biomarkers for disease. So we can identify subject and region-specific physiological response functions, like you can see here for the respiratory response function. It might be very different in different parts of the brain. One can move to calibrated bold to identify physiological effects. And one can also look at the coupling of neuronal processes for physiological regulation deliberately. You might ask yourself, is there even a resemblance of the neuronal networks or neuronal architecture and the vascular network architecture? In conclusion, denoising disentangles functional signal from much larger fluctuations in the brain to improve sensitivity and specificity of fMRI analyses. Noise generators are the MR scanner, subject motion, and physiology. And denoising as a process starts already with the acquisition choices and the experimental design. If you co-model signal and noise, or you reuse it, this allows for better interpretability than if you do it during pre-processing, so noise reduction. And finally, physiological noise can be relevant signal depending on your research question. And never forget, look at your data, signal and noise. With that, I would like to thank you and refer to the question and answers of the ISMRM for questions, or you can also write me on Twitter. Thank you to my lab, my previous labs, and my collaborators all over the world. And if you want to read more about physiological noise and denoising in general, here are some references, and the bold ones are very good reviews on different aspects of denoising.